Hello, everyone. Happy Wednesday. Welcome back to another installation of the Bodega Marine Laboratories seminar series. Thank you for being here. I'm very excited today to introduce Dr. Yui Takeshita. Dr. Takeshita is a scientist. I know it's a scientist at Monterey Bay Aquarium Research Institute in Bari, whose main research interests are to develop and apply autonomous sensing technology to observe marine biogeochemical cycles in situ. Yui graduated from UCSD's Scripps Institution of Oceanography and was a postdoctoral researcher at Carnegie Institution for Science in the Department of Global Ecology at Stanford with Ken Caldeira. And during that time, Yui collaborated with many researchers here at Bodega, working on everything from coral reef biogeochemistry to our local seagrass ecosystem. So lots of relationships that cross-cut our disciplines here at Bodega. So we're all excited to welcome him back here. Yui has received several honors, including the Excellence in Partnership Award from the National Oceanographic Partnership Program, or NOP, and the UCSD Director's Fellowship. So without further ado, the title of his talk today is Observing the Open to Coastal Ocean with Profiling Floats and Underwater Gliders. All right, let's welcome Yui.
Thank you, Dr. Alyssa. Okay, this is on, right? Well, thanks, Thanks for the introduction and thanks for having me back here. You know, I gave a seminar, I can't remember, 2017 or something like that. So I love this place. You know, like Alyssa said, I used to come up here, you know, to help out with and work on like the analytical systems, did some experiments in tide pools and whatnot. But that's not what I'm going to talk about today. I want to share work that we have been working on over the past many years, and as the title suggests, it's trying to establish a sustained network of a global ocean observing system consisting of profiling floats and gliders. And Alyssa here has this little Lego of a profiling float. If you don't know what it is, pass it around. We actually have one for a glider too, but I forgot to bring it. You know, I'm out of practice, but yeah, so that's about the right proportion. But anyways, that's, if you want one of those, just send me an email, and I'm happy to send them. But anyways, so this picture here is kind of like the dream, right? This is what we are working towards. This is what an established array of 1,000 biogeochemical floats in yellow, and the red are 100 sustained transects or areas for underwater gliders. So this is, you can see the floats measure the open ocean, and then the gliders connect the open ocean to the coastal ocean. And so they're very synergistic, and we need different observing technologies to be able to observe different regions of the ocean because they have different requirements. So before I start this work, you know, I'm presenting on behalf of many people, many years, and many institutions and funding agencies. So the funding agencies on top, and then the institutions and program are on the bottom. But you know, I have the privilege of being able to share the results of many hard work. So it takes a village. It takes a city, I think, more than a village. Okay, so we all know, so why do we care about observing the ocean, right? So we all know that the ocean is changing. Human-induced climate change has multiple impacts. And so here are 4 changes in the ocean that I think many people here have heard about. And we are interested in understanding the impacts of marine heat waves, ocean acidification, ocean deoxygenation, and changes in productivity due to changes in nutrient distribution, right? So these affect various the phytoplankton at the bottom of the food web, or higher trophic levels for ocean acidification and deoxygenation. So these are the global stressors that we know that oceans are going through, right? And then so as a society, we really want to understand how they're changing, how fast they're changing, and then ultimately what the impacts on the ecosystem are. And then so, and we have many tools to try to observe these changes, right? And then so for primary production, our best tool we have right now is satellites. So this is a map of the ocean NPP, net primary productivity, calculated from satellites. So you can see the red areas; these are high productivity areas, and lower in blue in the oligotrophic gyres. So these satellites create these beautiful maps every 8 days, global coverage. And with PACE coming online, we're going to be able to get more and more information. But if you peel the onion just a little bit, this is a data set that goes into constraining and calculating net primary productivity. Right, so each one of these dots, there's, I believe, an incubation of calculating or measuring what NPP was in a bottle. You measure biooptical properties, and then you use the satellite measurements to then calculate what NPP is. But you can notice some immediate problems. There are many places in the oceans where these algorithms are completely unconstrained. There's no data behind them, right? And so you have to interpolate between all of these data sets or these data points that go into training these algorithms. CO2 flux, we obviously care about how much CO2 the ocean is absorbing, right? And then so there are a lot of mapping techniques and products out there. So this is from Landschutzer et al. 2014. Red areas are where CO2 is outgassing from the ocean, and blue is where CO2 is going into the ocean. And so beautiful maps, they have monthly resolved, I believe, maps, of these CO2 fluxes based on a neural network approach. But again, what data goes into making these maps? And for CO2 fluxes, they rely on the SOCAT data product, the Surface Ocean Carbon Dioxide Atlas. And so this is a map of all of the data that goes into SOCAT. And then so looking at that looks pretty good, right? And then so there's a good coverage in the Northern Hemisphere. There's some gaps in the Southern Ocean, but it looks like there's global coverage. So that'll give you, that gives you, that makes you feel good. But if you look at how much data goes into these products every single year, this is from 2017, 2018, 2019, and 2020, and 2021. We all know what happened in the 2020, 2021, and data coverage went down. But even before then, in the best of years, there are still many parts of the ocean that are not measured, right, in the Southern Hemisphere. And many of these transects are only occupied once a year. And then so now, all of a sudden, the data coverage doesn't look quite as dense as a compiled data set that is collected over close to a century now, from, not really, but 1970 or something like that. So if you want to look at interannual changes, how El Nino is affecting how CO2 is going into the ocean, how the CO2 fluxes are changing, from year to year, these data sets are, we're relying a lot on the algorithms to interpolate accurately and project how the CO2 fluxes are changing, and in the future. And finally, and right now when the oceans are changing the most, our ability to observe the ocean is going down as well. So this is a plot of the number of oxygen profiles at least 1,000 meters depth in the World Ocean Database. So this is where, all of the, research crew's data are supposed to get archived. And then so, shipboard, there's glory days in the 80s. Now this is where the most shipboard measurements happen. And then, but you can see this continuous decline, over the past 4 decades of shipboard observations. These are oxygen measurements, but this is the same for all the other, other biogeochemical parameters of nitrate, pH, and things like that. So when we need observations the most, we are making fewer and fewer observations to observe these changes, and it's really, hampering our ability to predict and measure and kind of project out what the changes and the impacts of the changes will be into the future. So this is, coming back to the title slide, the solution to this undersampling problem is an array of robotic platforms that are out there all the time, measuring these key biogeochemical parameters, and then sending the data back in real time. So that if we can get a global coverage, then we don't have these, we can get it, even density measurements across the globe, monthly, annually, interannually, and that gives us a lot more ability to be able to measure and predict these changes. And then so first, I'll talk about profiling floats. That's the Lego that pass around. Thank you, Alyssa. So who here has heard of the Argo program and or knows what the profiling float is? Perfect. Okay, so profiling float, it looks like this, right? It's about the size of a gas cylinder. And so what it does, it is a profiling flow. So it profiles, so it goes up and down the water column. And how it does that is by changing its density. Right, so a denser object sinks, and then less dense object floats. And then so how a float changes its density is that there is a reservoir of oil, right? And there's an external bladder. And then so you can push the oil out into the external bladder. It's like a balloon, so the balloon will expand, right? So the overall mass of the float doesn't change, but the overall volume does, right? And density is mass over volume. And then so that changes the density. And then if you retract the oil back into the float, then your mass, again, stays the same, but your volume changes. So you can change the density of the float by just pumping oil in and out of this reservoir, and that allows the float to go up and down. And then so this is a picture of a non-inflated bladder at the bottom of the float. And then just a guess, so about a gas cylinder, how much oil do you think you need to pump out of the float to go down to 2,000 meters? A gallon. It's actually a cup, right? So this is an inflated bladder, barely noticeable, 260 milliliters. It's a little over a cup. But you only have to pump that much oil to change the density of a float so they can go from the surface to 2,000 meters, right? So we think of, physical oceanographers, they all tell us the ocean is very stratified, but ocean's not that stratified if you, sorry, you know what I mean. You don't need to change the density by that much, right? And then what that means is that you don't have to spend a lot of energy to be able to change the density for the float to go from the surface all the way to 2,000 meters. So this is an extremely efficient technology that allows us to collect data from the surface to 2,000 meters deep. And so then the typical Argo mission, Argo is the program that has this network of these profiling floats. So you deploy a float, it goes down to 1,000 meters, it drifts for 10 days, and then it goes down to 2,000 meters, comes back up, and as it comes back up, it collects measurements, so the core Argo arrays, temperature and salinity. It sends data back via satellite, and then goes back down to 1,000 meters, and then just keeps floating for 10 days and repeats that over and over again. The data is made publicly available within typically 6 hours of collection, most times faster. And then anyone can use that, no strings attached. Their automated quality control that's applied, and then once a year, the data is then, delayed mode quality control, which means that it goes to additional quality control to provide climate-ready data. And so this is an array of, I guess, is this today or yesterday? I can't remember. Of a map of the Argo floats in the world right now. So each one of these black dots, one of those profiling floats has surfaced and sent data back in the last 10 days. Right, so every 10 days, one of these dots comes back up to the surface and sends data. So then you can see how much better of coverage we have from the Argo program compared to the previous examples I've given. And again, I want to emphasize that this data is freely available in real time, and that has been, I think, one of the key successes of Argo, is that, you deploy the floats, quality control the data, but anyone can use it, right? So thousands of papers have been written on it. It's used for operational weather forecasts, fisheries models, climate models, and all of that. So it's been truly a successful program in oceanography, and it has revolutionized how physical oceanographers understand the ocean. And to kind of give you an example, so this is the density of measurements of temperature down to at least 1,000 meters, I believe, over 100 years. So the warmer colors, the purple and reds, are higher density than the cooler colors. And as you can see, again, Northern Hemisphere has better coverage, and the white, but there's still parts of the ocean that ships have never measured, right? And this is what Argo did in 20 years. It has really made the global map purple. So that really allows us to understand how much the ocean is warming. We know that the ocean is absorbing 90% of the excess heat because of Argo, right? Rain or shine, vacation or not, under sea ice or not, these profiling floats are working hard, going up and down collecting data for us. So we get an evenly spatially distributed data set, and that's not aliased in space or time, right? We get measurements every 10 days. And these maps, I think, are available on the Argo website, so I highly recommend people to go check that out. And I think this map, or this plot really blows my mind. This is just the global average ocean temperature anomaly, right? So this is the average temperature. I think it's referenced to 2014 or 2013, and that's how much the temperature has changed, global average. And I believe it's just literally mean Argo temperature, that's it, right? And then you can see this clear increase in temperature of about 0.3 degrees in the surface ocean. We can see that also the ocean is warming all the way down to 2,000 meters. And you can never make this plot, or this accurate of a plot with ships. You can only do this because floats are everywhere. And it's evenly measuring over time too. And so that gives you, so there are parts there's warming and then cooling, El Nino effects and all that, but this is unequivocal evidence, right, that the ocean is warming. And we can get the rate of it. Yeah. Oh, I know you're the physicalist, not. Well, there's deep ocean, like circulation. So like the deep water formation area, so the North Atlantic, Southern Ocean. Southern Ocean absorbs a lot of the heat, right? And then so when bottom waters, so heat is absorbed at the surface, and then heat gets transported down through the thermohaline circulation, right? Get that right? Did I pass my quals exam? Yeah, no, absolutely. And this cuts off at 2,000 meters because that's how deep the floats go, but you can imagine the heat's actually getting deeper than 2,000 meters, right? And that's why one branch is called Deep Argo. They want to measure the heat budget all the way to the bottom of the ocean. So, but that's a talk for another time. Okay, so that's core Argo. That measures temperature salinity. And what we are working on is BGC Argo, so biogeochemical Argo. And so I hope I've convinced you how successful and how effective of a program Argo is. And then so if we can add additional sensors onto these profiling floats, we can measure more ocean change. And so biogeochemical Argo, and now there are a suite of 6 commercially available, robust BGC sensors that can be equipped on profiling floats and operated for many, many years. So that's oxygen, nitrate, pH. So pH gives you information about carbon dioxide. So that O, N, and C, that forms your fundamental elements of life. Chlorophyll A, fluorescence. Optical backscatter. So optical backscatter is, like when you're driving in fog and you turn on your high beams, a lot of light comes back, right? That's the same idea. You flash light and see how much light comes back. And that's proportional to how many particles are in the water. And in the open ocean, that's a really good proxy for particulate organic carbon for phytoplankton and biomass. And the last one is irradiance, so downwelling irradiance, so how much light penetrates throughout the water column. And so there are multiple flavors of commercially available profiling floats, the Navis, Apex, Provor, the BGC Solo 2. All of them can be equipped with all six of these biogeochemical sensors. The oxygen, I'm not going to go into the details, but I'm happy to if you ask me later. Oxygen is measured using optical sensors. We have products from Ondera and Seabird, RBR. Nitrate is measured by UV absorbance, and then so it's ISIS, the in situ ultraviolet spectrophotometer. And so this technology was invented at MBARI and licensed to Seabird. The pH is measured by ISPET pH sensor, the ion-sensitive field effect transistor, also invented at MBARI and licensed to Seabird. Chlorophyll fluorometer, optical backscatter are measured by a single sensor, 2 different channels, and the downwelling irradiance is located at the top of the float, and it looks something like that. So, and all of these products, floats, and sensors are commercially available, and that's a key part of creating a scalable and sustained network. So BGC Argo is a part of the One Argo. So what I showed you before was Core Argo. And then so the next phase of Argo is One Argo, where the target is now 4,700 floats, higher density in boundary currents and in the equatorial oceans, consisting of 2,500 core floats, so just measuring temperature and salinity, 1,200 deep floats, so the floats that go to 6,000 meters, and 1,000 biogeochemical floats. And so a lot of effort went into constraining and understanding how many BGC flows we would need to constrain CO2 fluxes, nutrient distributions, oxygen distributions, and things like that. So we didn't just randomly pick this number. There were modeling efforts and stuff that went into it. And then there is a biogeochemical Argo science and implementation plan. We're in the process of updating it right now, but if you're interested in how we got to this 1,000 float number and various other science applications that can be done with BGC floats, I highly recommend you read this report. Yeah, so we've been working on this effort for many years, as I mentioned, and by we, the royal we. And then so there are two big projects that MBARI has been involved in, funded by the National Science Foundation. The first one is SOCOM, the Southern Ocean Carbon and Climate Observations and Modeling project. This started in 2014. It was led, the lead PI was Jorge Sarmiento at Princeton. And so this was funded to demonstrate, to deploy 200 BGC floats in the Southern Ocean. So the Southern Ocean is an extremely hard ocean basin to observe. So if you've ever done a cruise down there, you know what I'm talking about. There's 40-foot seas. No one goes down there in the winter because it's too scary. And so even the best of times, there's just not a lot of observations. But it absorbs the majority of the heat and CO2 in the ocean. So it's one of the, it's a really important ocean basin, but it was the least observed ocean basin in the world. But SOCOM, now we're in SOCOM 3, so the third renewal for SOCOM. We have deployed over 300, maybe not that much, 250, 300 profiling floats, BGC Argo floats through SOCOM. And this is a map of all of the floats that have been deployed and operated through the SOCOM project. And now, I think for many parameters, these biogeochemical parameters, the Southern Ocean has become from the least observed ocean basin to the best observed. Because of every single one of these floats is coming back every 10 days with new data that's never been collected before. Right, so this was kind of the feasibility project to see if we can really operate an array of floats in an ocean basin. And we could. And then so 5 years ago, I can't believe it's 5 years ago now, we wrote a proposal to the National Science Foundation to the Mid-Scale Research Infrastructure Program. So this is a new initiative by NSF. And they funded the Global Ocean Biogeochemical Array. And then so now SOCOM was a regional to basin scale array. GoBGC is going global. So we are funded to deploy 500 profiling floats globally. And so 500 is half of the target 1,000 float array. Historically, the US has deployed and maintained half of the Argo array. So GoBGC was funded to establish the US contributions to the global BGC Argo program. And so this is a map. The yellow ones are GoBGC floats, white ones are SOCOM floats. We have deployed 277 of the 500 floats, and we have a year and a half more to finish the rest, and we're on track to do that. But one thing that you can notice is that this array, this BGC Argo array, is really a global array now, right? And that's really exciting because once you have a global array, you can start to do a lot more cool science. And again, I will emphasize it takes a village, a huge village to do this. This is a picture of just the executive team for GoBGC. So, in each lab under each of these PIs, then there's dozens of people with it. It really is a huge number of people and effort went into making this possible. So I'm going to so this is the target. This is randomly placed 1,000 floats, what it would look like. And this is our current status. So yellow is the US and the blue is international. So we have over about 550 operational floats that have at least 4 of those BGC sensors, right? And then a big chunk of that global array is funded, and deployed through SOCOM and GoBGC. And as a result, so now this is the same plot. This is the number of profiles to at least 1,000 meters from ships before I showed, and this is now what floats are collecting. You can see for oxygen, floats now are the dominant source of oxygen profiles in the global ocean, right? By couple orders of magnitude. And that's now similar that's true for other parameters as well. So this is oxygen, orange is pH, pink is nitrate, and green is chlorophyll fluorescence. And so these are, again, data from the World Ocean Database down to 1,000 meters. And as you can see, around 2000, oxygen was first, chlorophyll, and then nitrate, and then pH. And then so as these global arrays are established, we are collecting more and more profiles of all of these parameters. And as you can see, as a number of shipboard measurements. So we really are collecting, these flows are collecting a lot of data. And then so your natural next question would be, I think, how good is the data, right? Sensor data is awesome, but how accurate is it? So we have put in a lot of effort into validating the sensor measurements from these floats. So these are results updated from Maurer et al. 2021. So we do our best to deploy a float from research vessels, in particular BP hydrography cruises like GoShip, where they collect the highest quality shipboard measurements available. And then so whenever they deploy a float, they take a CTD cast next to it. And then so through that we can validate the sensor measurements. So the float measurements are on the y-axis for oxygen, nitrate, and pH. And then the shipboard measurements are on the x-axis. And you can see, we have over 2,000 comparisons for oxygen, nitrate, and about 1,000 for pH. And these are collected globally. And with, if you look at the mean offset and the standard deviation, we can show that oxygen is accurate to 1%, or about 3 micromole per kilogram, nitrate to about half a micromole per kilogram, and pH is about 0.007. So not quite as good as the shipboard measurements, but pretty close, but our strength is that we can collect a lot more data. So we can fill in the measurements in between the shipboard measurements that can only go out there once a decade or something like that. Okay, so what science can be done from this array? And then so so there was a seminal paper, Johnson et al. 2009, that came out of a OCB workshop that envisioned what a global array of profiling floats and gliders could look like, right? And when they had this workshop, this is what the array of BGC floats look like. So oxygen in red, nitrate in orange, so they were just getting deployed, and green are the chlorophyll fluorescent floats. And then so this era, initially, the science that was done with these floats really were single to several float perspectives, right? So a research group got funding, deployed a float or 2 or 5, and then they wrote a couple papers on that, right? But then when you go to a global array, now you can do synthesis papers. You don't have to take single float perspectives. You can actually aggregate all the float data that's out there and then synthesize it. And then so that I kind of think of that as the regional to global array perspective. So the science that can be done with these floats have really shifted over the past couple decades as well. And I kind of give you an example, a classic one, productivity, right? How much productivity is happening in the ocean. And so this was the first set of oxygen floats that were deployed in Hawaii, science paper, Advisor and Johnson. And then they showed that near Station Aloha, the ocean was net autotrophic. And then so this is a plot of oxygen each year. So during the summer, there's a mixed layer here, mixes wintertime deep mixing, and then as the water stratifies, oxygen accumulates here. Right? So this is called a shallow oxygen maximum zone. And so because there's enough light to promote photosynthesis, but the mixed layer is only 50 meters deep, the oxygen accumulates here. So then by measuring the change in oxygen in this shallow oxygen maximum layer, you can calculate how much photosynthesis must have been there in order to get this change in oxygen. Right? And then so there was this debate going on. If you look at all the bottle incubation data, it suggests that oligotrophic gyres are net heterotrophic, which means that there has to be an input of additional carbon into these ecosystems. But a lot of these geochemical approaches are suggesting that trophic gyres are actually autotrophic. They're producing more carbon than they're respiring. And so this is very strong evidence, right, that it is net autotrophic. But this is, again, a single float, or these are 2 floats, so several float perspective. And this is an increase in oxygen in those layers that I was talking about. But now if you have a global array of oxygen, so this is a couple years old now, that was just measuring the productivity at Hawaii, but now we can get global primary productivity. And how this does this, I love this. So you can actually capture the diel cycle of oxygen from these floats, right? And so diel cycle, so this is daily cycle. This is again from Hawaii here, but a float comes up every 10 days, so you might be asking yourself, how do you get a composite like this? And you have to remember, if there's a lot of floats, some floats come up at 2 a.m., some floats come up at 4 a.m., some floats come up at 10 p.m., and all that. So if there's enough floats in a region that come up at randomly different times, you can aggregate all that data and put it in to create this composite diel cycle, right? And then so during the nighttime, there's respiration, so the oxygen goes down a little bit. We're talking 1 micromole per kilogram. During the day, there's net photosynthesis, so oxygen goes up. And then at night again, it goes down. Right? So then you can calculate how much productivity is happening in the ocean by looking at the amplitude of this diel cycle. Right? And then so, Johnson and Biff did this, plotted the productivity throughout the whole global ocean, by looking at all of these floats. And then now we can update this. There's a lot more floats now, so now we can actually look at so they just made annual primary productivity rates, but you can start to look at seasonal, so interannual changes and variability in global primary productivity. So we don't have to rely only on satellites now. We have a second way to constrain global primary productivity. Another approach to measure net community production is by looking at how much nitrate is drawn down from the ocean. So Guillaume Lineger, he's a postdoc at MBARI. So he took all of the nitrate data from the SOCOM project, shown here. He trained a neural network and then mapped it all out, mapped it a monthly map from 30 South all the way to Antarctica. And so this is a map of summertime nitrate and then wintertime nitrate. And then the difference between summer and winter, so during the growth phase, I'll tell you how much production happened during that season, right? Because the rec fuel ratio, right, the phytoplankton will uptake nitrate to grow. So if you measure how much nitrate disappeared in your growth season, then you know how much phytoplankton must have grown. And then so you can make these now maps of the mean annual net community production across the whole Southern Ocean. But since we have float data, monthly from 2014 to now, we can actually look at trends as well. So what Guillaume showed in his paper, that's coming out soon, is that the net community production is increasing in the Southern Ocean, right? And then it has increased by about 5% over the last 2 decades. And this is in agreement with satellites that suggested that there is greening in the Southern Ocean as well. And now there is another independent way and method to demonstrate that, yes, I think that trend is real. And then so now we're trying to understand what's driving these trends. Is it changes in winds, changes in temperature, changes in ion flux? It's by understanding this change in, this really important ocean basin. We can do that with floats now. Finally, another example of synthesizing data. Individual float data are really hard to use, right? So what's really useful for the community are gridded products. And so this is a beautiful example, Jonathan Sharp et al. 2023, the GoBy-O2. So they created a global map of oxygen 1 by 1, gridded from the surface to, I think, 2,000 meters depth. So this is an animation showing the mean oxygen concentration at 200 decibars at monthly resolution. You can see the oxygen minimum zones here, Indian Ocean, Eastern Tropical, Pacific, Northeast Pacific as well. And you can see how the OMZs kind of change, month to month, year to year. And now, so we have this global gridded product. So we can do something very similar to the initial temperature plot that we showed you. We can look at oxygen inventories. Right, so how many, I think this is in teramoles or something like that, of oxygen is in the ocean, right? So this is 200 to 1,000 decibars. This is what global ocean circulation model suggests, right? So this is what the tool we had to suggest that the ocean is losing oxygen. But now this gridded product here, now constrained by observations, we show a very similar trend of declining oxygen global inventory from 2004 to 2022. So that's oxygen. And then so I think we are able to do this with carbon as well. So Guillaume also trained a neural network to synthesize all of the pH data from the SOCOM array. And so this is a change in DIC, the inventory, from 0 to 200 meters, 0 to 500 meters, 0 to 1,000, and 0 to 1,500 meters, right? And then, so this is, but you can see at all depth, DIC, so DIC is the total amount of carbon dioxide in seawater that is increasing at all depths. And maybe we would expect that because you know that the Southern Ocean is absorbing anthropogenic carbon dioxide and it's getting accumulated in these different layers. And then the rate, the deeper you integrate, so from the upper 200 meters, it's about 0.2 petagrams of carbon per year, 0.4, 0.6, 0.7 petagrams of carbon per year. And if you assume that this increase in DIC is due to anthropogenic carbon, then we can see this is the increase in your anthropogenic carbon inventory in the Southern Ocean. And if you compare these with other independent estimates, so Hawk et al. 2023, the ReCap project estimated the Southern Ocean carbon dioxide anthropogenic carbon inventory changes. They suggested 0.75 petagram per year. This is how much the Southern Ocean is absorbing CO2. Right? And this is through global ocean circulation models. If you use a surface PCO2 flux models, they get about 0.74. So the float-based estimates are pretty close to that. And they go down to the bottom of the ocean, so some of this difference is the fact that we only go to 1,500. But this is, this is awesome. We are actually doing carbon inventories with autonomous floats. Right? And this is important for people who are interested in MCDR. If you're gonna start mucking around with the ocean and try to get the ocean to capture more CO2, each company is gonna have, okay, we captured X kilogram tons of CO2, we did this, whatever. We can be the referee of the global ocean saying, well, this is how much carbon went into the ocean for this period, from month to month, year to year. And if all of those, companies, they don't add up to the total, then we know there was an accounting error there, right? So we can have the global account, we can be the global accounting system for, for MCDR and even just, climate, just additional anthropogenic carbon. Okay. We also have worked on making tools that make accessing the BGC Argo data straightforward. This is a tool developed by PMEL, the One Argo Toolbox. I'm going through this quickly because I'm running out of time. WebODB, so whoever has used Ocean Data View, now there's a web version of it. You just go to this link, make an account, and then the global BGC Argo dataset is uploaded and updated daily. And so you can start, the interface is identical to your desktop version. And you can just start playing around. You can extract data into a simple CSV format, and off you go. So, I highly recommend WebODB in class, for example. Okay, gliders. I have 5 more minutes, so I'm going to go through this pretty quickly. Okay, so gliders are, you can basically profile floats on a side with wings. So it still uses the same idea, same concept to go up and down the water, but they have wings, so that provides lift. So that moves them in the horizontal direction. So they dive kind of in this, V shape, right? So what that allows you, and then you can also steer them. So what that allows you to do is you get to navigate them, or you get to tell them where to go. So they're really, one really useful application for gliders is that they can just do transects, go back and forth in the same patch of water. And this is really good at saying how this transect here has changed over time if you keep occupying that transect, right? And they're particularly useful for the coastal ocean. This is a map of the residence time of water in various different coastal oceans around the world. So some places, these light blues are about 30 to 60, 90 days. Some regions are a couple of years, right? And then so the Argo program can constrain what's changing in the open ocean, but if that water is getting retained for years, that's not going to be exactly the same as what's happening in the open ocean. So then these gliders, by going from the coast to the open ocean and back, you can actually, it's a really powerful tool to observe changes in the coastal ocean and then connect the open ocean to coastal ocean dynamics. There have been, obviously a lot of BGC sensors put on these gliders as well. This is the types of BGC sensors that have been equipped on gliders. That's in the IU's Glider Data Assembly Center. But you can see oxygen, chlorophyll, fluorescence, backscatter. These are these matured sensors that are available for the community. There's a lot, 50, 60% of the gliders have them already. But when you get to pH and nitrate and light, there's a lot fewer gliders that have pH and nitrate sensors on them. So that's part of our lab's work. We're trying to develop pH and nitrate sensors that can be using the same sensing principles as BGC Argo floats, but then can be operated on gliders. And so our first attempt at this for pH, this is a pH sensor. We just took the exact same pH sensor that was on a float, made an adapter, pumped it, plumbed it into the pump flow stream of a spray glider. So we conducted multiple missions. This is Monterey Bay and Mbari's here. So these are the transects that I was talking about. So just gliders go back and forth. And then what you can get from these data are these section plots. So this is, the color is pH, and lower pH at depth, higher pH at the surface, as you would expect. And the black line here shows the saturation horizon for magnetite, right? So this is where equals 1, the magical numbers that things start dissolving in. So, but you can see, so this was during non-upwelling conditions here. The saturation horizon's pretty deep, about 100 meters. But then, in July, when there was strong upwelling winds, the saturation horizon in the bay comes up to about 10, 20 meters. And so you can see, and then you can see, this is the undercurrent, California undercurrent, that a lot of, mesoscale variability. And you can't capture this with ships. You really need, gliders that make measurements every couple kilometers. And you can see all this patchiness in here. So this is the type of data, one type of data that these gliders can collect. But in order to scale it, there are some platform limitations. The gliders that we use, the Spray 1 gliders, they're out of space, basically. This is what the wet bay looks like. We can't add any more sensors. Some groups have just strapped sensors onto the exterior of a glider, but that really increases drag and makes them hard to navigate and to settle on them. So Scripps, the instrument development group at Scripps, has developed the Spray 2 glider. It looks very similar to the Spray 1, but it's about 70% bigger. So it can hold a lot more sensors. And the controllers and the power communication systems were designed so that all 6 BGC parameters, oxygen, chlorophyll, fluorescence, backscatter, light, pH, and nitrate, can all be operated by this platform. And here's a picture of prototype 201, the first one that was deployed earlier this year. It's operating well. It's up to 203 or 4 at IDG now. And this is a pH sensor, so we modified what the pH sensor we use on these floats, on these gliders. So we made it modular. So basically, we apply power and you just get data back from it. So it makes it a modular design that makes it much more straightforward to integrate onto a variety of platforms. And then here's a picture of it, it's somewhere in there, I don't know where it is. Actually, I think it's right here. So that's the wet bay of the Spray 2. And the first pH glider mission by Scripps is designed, they're planning to deploy it in mid-May. So in a couple of weeks, we should get pH measurements from these gliders along our coasts. And Scripps operates a network of glider lines. So this is the California Underwater Glider Network. So there's one down here at Dana Point, Point Conception, the Monterey Bay, and now up here in Bodega. I believe Bodega operates, deploys, and recovers these gliders. Thank you very much. And so these 3 main lines have been going on since 2007, and these 2 new lines were added in, I think, 2020 or something like that. And then so we have funding to add 6 pH and nitrate sensors on these lines over the next couple of years. So the first one, I think, is going to be the southernmost line, but we are building 6 more pH sensors this year so that as these gliders become updated with Spray 2 gliders, they will become a part of these sustained transects here, right? So this is really helpful for us, the pH sensors that we build and calibrate in our lab. We can immediately use it by collaborating with these, with other groups that have sustained observational networks. So that's the fastest way that our technology can get used, make an impact in the ocean. And we also have, that's the West Coast, and we actually have funded projects to put pH and nitrate sensors in the East Coast as well. So this is the Gulfstream network operated by Robert Todd. And over the next couple of years, we're going to deploy full pH and nitrate sensors for them. So we're kind of starting to get at, sustained networks of biogeochemical sensors and from gliders and floats. So this is kind of the first step, and we're trying to establish this in the US. And so if you invite me in a couple of years, I can give you results from these networks. I'm going to, this is my last couple slides. So once you have all this data, you still need to synthesize it, right? And the coastal ocean is very dynamic, so techniques that work in the open ocean don't necessarily work in the coastal ocean. So the most powerful way to kind of assimilate and synthesize all this data is through models, ocean models, right? So ocean models can assimilate data, and then so they basically run their model, and then they compare their results with satellite observations, glider observations, nudge their models towards reality, and then so that they don't completely fly off the rails, essentially. So the more data you have to constrain really important parameters that you care about, the better your model is going to be. And then so we are starting a collaboration with the Ocean Modeling Group at UC Santa Cruz. And so they have the ROMS model, the Region Ocean Model for the California Current System. This is their domain. And they have started assimilating the pH data from our gliders. So one run in 2019, so that was the transect that they assimilated. And so this plot here shows the difference between the model and the glider. And this is before they assimilated the glider data. Right, so you can see in the near shore, the model is about 0.2, 0.3 pH higher than the observations. And this is where we care about it the most, right? People who study benthic, upwelling dynamics, benthic ecosystem impacts on ocean acidification and all that. If the models are off by 0.3, so that's not really useful, right? But once you assimilate the glider data, as you would expect along this transect, the model now agrees with observations to 0 by definition, right? But the thing that was interesting to me and us, everyone, was that by assimilating this one transect in April, this is the change in the surface pH across the whole domain. And I think this is an annual average. But you can see by changing the pH here in this little region, pH at the surface of Point Conception is changing by 0.1. Right? And this is because the ocean is transporting waters from, the undercurrent going north, California current going south. And so if you change properties up here, it has a large spatial impact. Right? So this is a huge benefit of using these models, is that by constraining parts of the model system, that you can actually improve the whole model region. And then so, we're working on assimilating, as the glider pH and nitrate data become available, they're going to start assimilating it and then try to see how, and then provide these gridded model outputs for anyone to use. So people, fishery folks can compare their fisheries yield to oxygen, changing pH, things like that. So we're working on synthesizing the data too. I'm going to skip this slide. Thank you.
That work? It did work. Awesome. Cool. Thank you so much for an awesome talk, Yui. I'm going to open up the floor for questions. We have plenty of time for questions. Yeah. Don't make me throw it. I hate throwing it. It gives me so much anxiety. Not as much anxiety.
Thank you. Great. For the floats that are in the open ocean. They go up and down. How do you ever control for horizontal movement? And so do they get like swept in currents? How do you reposition them if you need to?
So the answer is that we don't control them. So certain parts, certain areas, so the Gulf Stream, if you deploy in the Gulf Stream, they will just get kicked out in months. And some parts of the ocean are much more stable. Gulf of Alaska area, some floats would just stay in the same area for years. And so but the goal is to establish an evenly covered array, right? And so there's a whole team of people working on understanding what the probability of where the float will go if you deploy it, on this pixel versus that, and then so planning the array. So if you're like, okay, there's this whole certain emerging here, where are the optimal places to deploy it so that there's a higher probability that there will be coverage to deploy. Only time we pilot it is like if there's seamounts and it gets, sometimes it hits the ground, and then so you just profile it shallower.
I wanted to ask a question.
Oh.
Just a follow-up. A follow-up to that. Have there ever been, or how often are the times that photos float?
More often than you would want, I would say. So most of the institutions in the Argo program at the US institutions, they get the float or they build the float and then they open it back up, do a really thorough inspection, close it up, and then deploy it. And then so their chances of success goes up quite a bit. And so right now, I don't have the number of like survival per year, but in the core Argo array, about 50% of the floats reach about 80. And, so they have, if you think of, if there were no, unnatural deaths and all floats died because the battery was exhausted, battery in theory could last 9 or something. So some percentage of that dies before then. BGC floats are a little bit higher. There's more connectors, more. But right now, I think the aggregate. I think of it as like what the sensor failure rates are, because if the sensor fails, even if the float's going up and down, it's not collecting the sensor you want. Right? So the chance of all 6 sensors surviving per year is about 90%. But yeah.
Hey. Cool, this is super interesting. Woo. Yeah, I was also wondering about the lifetime of the floats and the gliders, and are there issues with fouling or yeah, I'm just thinking about some of like the like light sensing, how that's affected potentially by fouling, or are they moving fast enough or settling organisms can't catch them?
So I'll start with floats. One really beneficial aspect of the Argo program is that it sits for 10 days at 1,000 meters.
Yeah.
So it's like taking your sensor out of a refrigerator every 10 days, using it for 8 hours, and putting it back in. Not a lot of things tend to grow on floats. Gliders is a completely different, beast because they operate in coastal waters, so they typically won't dive shallower, and they're constantly, they're like a shark. If they stop moving, they die. And then so they're always diving. So 50% of the time, or some significant amount of the time, they're in the euphotic zone. And so light sensors, biofouling is going to be the biggest challenge. And so I don't have a good solution to that. Barnacles also really are a problem for gliders. We've had, we couldn't fly a glider properly because there's a big kelp, so biofouling for sure.
Yeah, I was wondering how fouling or age of floats also influence the accuracy of the. They tend to float over time, and you have multiple floats per period, check them against each other? Is that like,
We don't compare float to float, but we do, so different sensors have different ways that we adjust for sensor drift. All sensors drift. So oxygen sensors, we actually recalibrate them each time they come up to the surface because they can measure air, right? And then the partial pressure of oxygen is in air is constant, we know that. Calibrated that way. pH and nitrate, we have algorithms based on chipboard data that we can use to predict what pH and nitrate would be at 1,500 meters. So that's very low variability down there. And then so we adjust the float data to a reference field at depth, and then the rest of it, we rely on the sensors. But yeah, it's a tremendous amount of effort went into
yeah, sure.
developing those adjustments.
And the same time.
Temperature, we assume because there's no way to validate it, the misters don't drift. But salinity, yeah, it's similar. There's a lot more, there's now enough more, there's enough Argo floats now that you can actually use good Argo floats to, adjust the bad ones.
Yeah. Ready? Yeah.
I have kind of, I guess, a follow-up question, but like, good if that affects sensor backscatter, but
Sorry, what affects backscatter?
pollution, like runoff in.
I think it will, right? It will depend by if it contaminates, like it's still particles, right? And then so what the sensor gives you is a total particles attributed to water. And then so in the open ocean, most of that comes from phytoplankton. But when it goes into river mouths and stuff, for sure, you can't use it anymore. But floats can only operate where it's at least 2,000 meters. So there's not a lot of, most of the precipitation happens much more coastal, so it's not an issue.
You touched on the marine carbon dioxide, Southern Ocean, about calculating carbon budget and able to tell whether removal has reached the input that is estimated from the census. Do you have a sense of how those data might or thinking about, I even a small amount of removal would be better than nothing.
So MCDR signal, it gets so quickly, right? So we have to get to a certain scale before anything is going to be detectable from these kind of, even using the best shipboard analytical. So, and I don't know what the number is, right? But like the magic number that a lot of companies are shooting for is a gigaton per year, right? If all of the MCDR kind of adds up to a gigaton per year, then we will start to make a dent. So, that's about 10% of global emissions. But once it gets to that point, sustained for a couple of years, we should start to see a change in the global carbon budget. And so until we get to that point, that was, but to make that plot, it took 10 years of data, right? And all those floats. So you need to start creating these baselines before we start geoengineering, right? To be able to distinguish between the two processes. But what's also going to be really useful for, so my conversation with MCDR, MRV folks, really is that it's going to come down to models to tell you what the additionality is, right? Because if you do an intervention, you need to have a control case and then the experiment, which is a real life case. And then so you can't do that with just observation because you have to do a lot, well, you can, but there's a lot of assumptions going into the control, right? Because once you throw alkalinity in the water, for example, you can no longer measure what that would have done if you didn't add alkalinity, right? So in order to get the counterfactual, the way to do that is models. That's kind of how the community is moving towards that, so you have to run a control, an experimental model, and different tells you the additionality. But then in order to get really local processes right in an estuary or something, they have to be really high resolution models, right? Which you're not gonna be able to expand a domain by very much because it's so computationally heavy. And then so in order for those models to be trustworthy at all, you're gonna really need good boundary conditions that come from lower resolution models. So I think that's where, you know, kind of the California Current System ROMs model are gonna be really helpful, is that they can provide much more realistic data constrained boundary conditions for the nested models that the MCDR package rely on to calculate the addition.
I- That or sure. Change. About
Kind of the, I don't know what, I forget what paradox, but like a lot of the pilot studies that MCDR companies do is relatively small scale, and it's extremely difficult to track specific patches of water in the ocean. So a lot, you know, you could just go 1 year, be like, all right, we're just going to dump a ton of alkalinity, see what happens, because then we can actually measure it. But then the impacts, any unintended consequences are much higher, right? So, you know, by like we're doing this intermediate stage is actually the hardest. So that's kind of, you know.
All right, I think we're at time, but I do want to ask you one more question on behalf of my undergraduate students, who every time I talk about Argo and the Argo array, inevitably a student asks me every year, what happens when Argo floats die and where do they go?
Yeah, so yeah, we get that question a lot as well. And so the answer is that, so most likely, so the biggest draw to the battery is when they try to go from 1,000 meters to 2,000 meters, because they're sitting in the refrigerator and, you know, the battery is kind of, they've passivated. And so they try to move the hydraulic pump, they can't. Eventually run out of battery and just float there and then eventually implode and sink to the bottom. So we don't recover them. You know, that's an unfortunate, you know, that's the unfortunate reality of it. But I do think that it's important to, everything comes with its cost benefit analysis, right? And then what is the data that we get from these floats worth, you know, the floats that we're leaving in the ocean. And to kind of give you a perspective of the magnitude of the problem, you can fit all of the floats that the US deploys in 2 shipping containers. That's amount, that and that, you know, so that's, you know, in trade, I don't know, I've never been able to dig up how many shipping containers are lost, you know, but I guarantee it's a lot more than 2, right? And so, you know, I think it is just important to keep that in mind. And then also, if you wanted to go and get it, then it's going to cost a lot more in terms of CO2 emissions through the fuels from the ship and all other pollution that are associated with it to go get it, because floats are really, really small and the ocean's really, really big. So it's not practical to collect them, refurbish them, and then redeploy them. So.
Cool, I'm glad I'm telling them the same thing. Awesome. All right, let's thank Yui one more time. And we'll see everyone again next